Hello everybody, my name is Andrey Prjibelski and I work in St. Petersburg and Pavel Pevzner's lab. And today I'm going to present an ExSPAnder algorithm, which is a part of Spades genome assembler. As you may know, Spades is a bacterial genome assembler for short reads. But why do we even talk about short read assembly? Are there any challenges left there? Well, I would say yes. First question is that big biotechnological companies recently release a lot of new protocols and technologies, but do assemblers keep up with these advances? And the second question, can you assemble just a small bacterial genome with a short read library? Well, I would like to answer these questions at the end of my presentation, so at least some of you will listen till the end. Let's start from some basic stuff. We have a genome and we sequence it into short reads. And the question, how we can assemble it back. And since we use short reads, we will use the Brewing Graph framework. And we assume that Spades has already cleaned all erroneous and chimeric edges from the graph. So we have clean and pure assembly graph. And our question is to resolve repeats. But how we can do that if reads are shorter than the repeat lengths? As most assemblers do, we're going to use read pairs. Read pairs are just two genomic fragments that are separated by the insert size. Basically, there are two types of read pair libraries. Paired-end libraries with rather short insert size, and jumping libraries, or often called mate pair libraries, with insert size up to 20 kilobase pairs. The problem is that the insert size is not fixed. And different libraries have pretty different insert size distribution. If you look here at, at the distribution for paired-end libraries, we see a pretty nice distribution. Looks similar to normal one with a rather low deviation. But if we take a look at the insert size distribution for mate pair libraries, they can be pretty nasty. We have a lot of, Okay, as you see, we have a lot of read pairs with wrong orientation, and only a few correct read pairs. And corrected pairs have a pretty high deviation, which complicates the assembly. When we assemble a genome, we actually do not know these distributions. How we can obtain this distribution? We can align read pairs to the long edges of the Brewing Graph. And actually, we're going to be interested only in trusted read pairs that lie within the confidence interval. How we can apply read pairs to the assembly graph? We're going to construct paths. For example, we already have a path marked blue and several possible extension edges which are marked red. We're going to score each extension edge and select the best one. If we can find the best extension edge according to the score, we will extend the path P and stop it otherwise, and then we iterate the process. The decision rule is also pretty simple. If we have a certain extension with the score much larger than the score for any other extension, and if its score exceeds some small threshold, we're going to extend path P and stop otherwise. But most assemblies are currently done with several libraries. So if we have several libraries, we're going to sort them according to the insert size and just use the shortest library which provides us an extension. If no library provides us a correct extension, we're going to stop growing path P. Now we have the only challenge left, how to design a proper scoring function. There are several challenges. As I already mentioned, insert size distributions can be pretty nasty, so we have to deal with them. As you know, Spades is designed also for single-cell datasets obtained with multiple displacement amplification that have quite uneven coverage depth. So our scoring function has to deal with that also. And the last challenge is chimeric read pairs, which are also typical for single-cell datasets and mate pair libraries. Usually these reads have pretty abnormal insert size, so they can complicate the genome assembly problem. Okay, let's have a look at a simple example of the Brewing Graph. We have a read pair aligning to edges P and E at positions X and Y. Okay, let's construct a rectangle from edges P and E. And now our read pair will be just simply a point within this rectangle with coordinates x and y. Pretty simple. Okay, if we have a set of ideal read pairs with exact insert size, they would form a 45-degree line in this rectangle. But as I mentioned, read pairs have variations. So points corresponding to real read pairs are usually scattered within the rectangle. And since we're interested only in trusted read pairs, we're going to use points only within the confidence strip. Okay, one more simple example. We have a path P and two possible extensions. And the first and the most intuitive idea how to design a scoring function is just to use the number of read pairs within the confidence strip. But as you see, edges can have different lengths. So long edges would always win because they have more points in corresponding rectangles. So no, that doesn't work. Okay, let's try to normalize the number of read pairs. The first and the most simple idea how to normalize the number of read pairs is just to divide total number of points by the area of the confidence strip. But, as you remember, insert size distributions are not uniform. We usually expect more read pairs in the center of this confidence strip than on the side. So let's do it more carefully. This approach does not work as well. Let's combine our knowledge about the insert size distribution and the confidence strip. We're going to construct a cylinder based on this insert size distribution, and the volume of this cylinder will be the number of expected read pairs if we assume that the coverage is uniform. So now we can design a better scoring function by dividing the actual number of reads by the number of expected read pairs. And we further will call this value at density. Well, this looks pretty good. But when we extend a path, we aim to score an extension edge relative to the whole path, not just a single edge. How we can do that? Okay, construct a simple rectangle from a single edge to extension edge, attach a new one and a new one. We have a composite rectangle that consists of three simple rectangles. And we can do the similar for edge E2. In this example, you can see green lines which correspond to the real genome that we do not know. And our goal is to detect the correct extension. If we look at these rectangles, you see here the density for the correct extension will be higher than the density for incorrect. So our algorithm will choose correct extension E1. But as you remember, we talked about single cell datasets with uneven coverage. And we can imagine a situation when the upper path has low coverage and the lower path has high coverage. In this case, it may appear that correct extension has pretty low density just because the number of read pairs is low. And the incorrect extension has high density just because the coverage is high. So this approach is not working as well. Let's consider each rectangle separately. We say that a simple rectangle, for example P1, E1, is good if its density exceeds some predefined threshold. And we can define a new scoring function, which actually the number of good rectangles relatively to the number of all rectangles. So for correct extension, we have all three rectangles which are good. So the score will be 1. And here we'll have score only 2/3 because the middle rectangle is bad. But as you remember, edges have different lengths, and thus rectangles have different sizes. And it's not good to assign an equal weight to all of them. So let's try something more intelligent. Instead of assigning an equal weight for each rectangle, we will assign different weights. And we have such function, the expected number of read pairs. So the final scoring function will be just the number of expected read pairs in good rectangles divided by the total number of expected read pairs. So we will get 1 here and approximately 0.5 for incorrect extension. Okay, and the last algorithmic part for today. How we do with long repeats. We have a path in the Brewing Graph, and R is a long repetitive edge. When we look at the rectangles corresponding to extension edges, you see that the scores are pretty similar. 1.0 for correct extension and 0.9 for incorrect. and our algorithm cannot distinguish which one is correct and which one is not. But if we look closer, we see that read pairs that connect repetitive edge with extension edges, they support both extensions. So actually, the repetitive edge does not provide any valuable information for us. Okay, let's ignore it. Once we ignore the repetitive edge, which has ambiguous read pairs, we see that the score for correct extension remains 1, and score for incorrect extension becomes 0. So we're sure that E is a correct extension. Okay, enough algorithms for today. Let's talk about something more interesting, for example, about the results. Okay, we benchmarked our assembler on many, many data sets, but of course I'm able to show only a couple of them. Here is a benchmark on single-cell data sets. To compare different assemblies, we used Quast tool, and we picked some important metrics as an N50, the longest scaffold, the number of breakpoints between the reference genome and scaffolds, and the percentage of genome recovered. As you see, we do pretty well on single-cell datasets comparing to such popular assemblies as ABIS, Ray, Velvet, IDB-AUD, and even AllPass. And when we talk about mate pairs, we assemble single-cell datasets into just 4 contigs, recovering more than 99% of the genome, and this is pretty high for single-cell dataset. We actually do pretty well on standard bacterial datasets. As you see, when assembling paired-end datasets, all assemblies do pretty well, but when it comes to mate pairs, Space can assemble a complete genome with no misassemblies, recovering 100% of bases. Okay, now let's return to the questions that I asked in the beginning. Do assemblers keep up with advances in sample preparation? Space does. We have different scaffolding algorithms for paired-end and mate pair libraries. We have hybrid assembly with PacBio and Sanger reads, and we also support both Illumina and IronTorrent short reads. And let's return to the Illumina Nextera mate pair library that I mentioned in the beginning. When we look at conventional mate pair libraries, they have pretty nasty distributions with a lot of read pairs with wrong orientation and only a small fraction of correct read pairs. But Illumina Nextera mate pair libraries look just a large paired-end library, just with a bit higher deviation. So that gives an answer for the second question. Can we assemble a complete bacterial genome with a single short-read library? Yes, we can. We tried to assemble Nextera mate pair libraries with different assemblers, and as you see, Soap, Nova, Velvet, and Space do pretty well assembling with very high N50, but many assemblers do a lot of misassemblies, and Space does only a few and recovers 100% of the genome. So what's the message here is actually you can do the assembly of similar quality to PacBio with short reads. This is maybe pretty amazing because just short reads with 200 bases long and similar quality to 5 kilobase PacBio reads. Okay, I guess this is enough for today. Oh, no, I forgot. If you think this is good, I would say that the things are even better because Space is not just good algorithms, it's pretty user-friendly, and you can find us on Illumina-based Space, DNA Nexus, Live Technology Storage Server, and Galaxy Toolshed. And if you wish to know more about our assembler, just come to our poster N10. And now that's all. I want to thank our funding, and I want to thank NSF for providing travel funding to ISMB. And I want to thank all our team, and especially the head of our lab, Pavel Pevsner, and I want to thank Irina Vasilynets, she's right there, because this is a joint work with her. And now it's over, and I would like to answer your questions, and thank you.